Hi, I'm just here to help you set up.
Thank you. Do you think it will be possible to use a laptop? Or...
Yes.
Yeah.
Is it a Mac though?
It's a Mac though. I have the adapter.
Do you have that? Yeah, if you have the adapter, that's great.
Let's try. Does it work on any of those?
Yes, it should.
Yeah. And do we need to log in?
I've done all of it, so you just have to.
Okay, so even with a laptop, I don't need to log in. By the laptop. Oh, that's cool. Just need to load Danika's talk. We have the country acknowledgement slide.
We have it usually in the hub.
Yeah, okay, no worries, I can pull it out. I should probably have loaded your talk first.
I'm
then
sorry.
then
Why is
just like
your
drop it in.
presentation
Yeah,
not coming up?
oh good. And we will look at this. Where's mine out there? Just change the resolution and
So we just have to figure out why your laptop's not coming up.
Is it not coming up?
Yes, so it says ensure that your laptop's set to output video for external display.
Set to.
Do you need to go full screen? Like, is that?
Does that help?
No, so it would come up otherwise. So you have to go to your display settings.
Display. Let's see. Where that might be. Appearance.
Displays.
Displays. Use
Says
as
external.
Stop moving. External. This one. This one. There's nothing I can change. Advanced, maybe.
No.
No. Okay. I don't mind use the PC, but we might want to have a look at.
I mean, it doesn't work. I'm just wondering if
because of the cable, maybe. I have it. I will pull it up. So what do? We can use the PC, maybe?
Yeah, if you can use the PC. Straight to me through your USB.
Yeah. What I want to do is to pull out the welcome country slide, which is here. I'll probably have it here already. CS, yeah. Okay, thank you. Mac never works.
What is this?
This
Yeah.
don't. Yeah. And so this one.
This one.
Okay.
This one, the second one.
Yeah. So all three of them.
I'm not sure what's happening.
Yeah, I can do this. Thank you. Because this one probably otherwise won't open.
Just yesterday. I know. That's the attitude you
Country acknowledgement.
jarring. That's how jarring. When I look in the mirror, I'm like, whoa.
Yeah. Just open everything, see whether that works. Yeah, it works. All good? Do you want to?
I mean, yeah, there will be slides that won't have dots, but that's fine. What am I going to do now?
My laptop can't be connected to the big screen. Okay, all good. And
Was
what?
that all three?
Yeah, I mean, this is the acknowledgement
Yeah.
of country, and then 2 talks. That's
Yeah,
it.
that's it.
So we don't need to share the screen or anything.
No, you
right?
don't
So
need to
just
do
play.
anything.
Okay.
Yeah.
So you've got both talks in the one presentation?
No, two.
We'll
two
just
of
exit
them.
it and then open.
Okay.
Yeah.
Who's going first?
Wait.
So approximately slightly less than 30 minutes. Questions within your 30 minutes.
Yeah, yeah.
So I'll put the motion at 20 or something.
Yes, please.
Wrap it up.
Yeah. Then they stop.
Yeah, exactly. Okay, well, it's about time to get started. We have a packed schedule with 2 of our outstanding EMCRs going to speak to us today. It's my pleasure as the head of the Department of Immunology to stand in for the acting head of the school, who's standing in for the head of the school, to welcome you to the seminar at the School of Translational Medicine. But first, or second, I'd like to acknowledge the Boonwurrung Wurundjeri clans of the Kulin Nation, who are the custodians of the lands and waters. We pay our respects to their elders, past and present. So it's a very great pleasure to me, as I indicated, to be able to introduce 2 speakers who I've had very close contact with over the last few years. First, we have Zhoujie, who will be Zhoujie Ding, a postdoctoral fellow in my group, who graduated with a PhD from Sweden in 2015, moved to Melbourne in 2018, and has been working assiduously on plasma cell biology in that time. Been extraordinarily successful and had lots of great papers, but recently took an excursion into a quite unusual field, I think, for us, unexpectedly. However, and fortuitously, it returned to B-cell development, so we all felt much relief and comfort at that. Many of you would know Zoe not only from her great work, but she's involved in so many of the activities of the school and the campus. She's one of the integral parts, I think, that keeps the whole thing as a collective and keeps it going. She's outstanding. She's with Immumon, which reaches to the larger campus, and the Equity, Diversity, Inclusion committees, and many other things which are too numerous to mention. So it's a great pleasure for me to welcome Zoe to talk on Ki-67, which is, it seems, more than just a proliferation marker. So Zoe, please.
Thank you, David, for the kind introduction. And yes, so I guess I don't need to introduce myself again. But for those who don't really know me, I am from Department of Immunology and from David's lab, the Immune Memory Laboratory. So what is immune memory? I mean, all of you here who from the Department of Immunology probably knows, but for the sake of the broader audience. So immune memory is the ability of the immune system that mount an immune response to foreign substance, but also to remember those challenges so that the subsequent a subsequent response can be a bit faster and better when our immune system sees those foreign substance. And antibodies are really important for our immune memory. And actually, that's the focus of our lab. And we are interested in the cells that are producing those antibodies called plasma cells. One of the key questions that our lab is researching on is what determines the lifespan of those antibody secreting cells. And through lots of few years of work, as you can see here, we started to uncover the mechanisms of plasma cell turnover, the factors regulating those cells. However, I'm not going to talk about plasma cells today, as David already hinted, but going to the beginning of those lymphocytes. So I'm going to talk about lymphopoiesis. And it's a protein that you're probably very familiar with, that's Ki-67. I guess anyone doing immunology or cancer work have used Ki-67 as a proliferation marker in your assays. But what do you know beyond the proliferation marker role of Ki-67? So a group in 2016 published a paper in Nature, and they are interested in what function of Ki-67 can play. And they found using HeLa cells and siRNA to knock down those Ki-67, that Ki-67 is important for maintaining chromosome structures during mitosis. And they work as brush coating those chromatins during mitosis. And in the follow-up study, they found that Ki-67 is also important when the cells are trying to exit those mitosis. So they promote chromosome clustering at the end of mitosis. At the same time as the first paper, another group found that Ki-67 is also required for maintaining heterochromatin during interphase. So basically, Ki-67 is required for any cells that's within the cell cycle. And other groups also found using cell lines that Ki-67 is required for normal gene expression, especially in cancer cell lines that they research on. So as you can see, those studies mainly using cell lines. So what are the roles of Ki-67 in primary cells, and what's their immunological function, which I'm interested in the most? To study that, our collaborator at Zhoujie, they made a Ki-67 knockout mouse model by deleting the exon 3 in the Ki-67 allele. And they validated the Ki-67 expression in liver cells by Western and in proliferating cells by flow cytometry. As you can see, our Ki-67 knockout mouse do not express the protein. So how is the animal itself then? So we found that those Ki67 knockout mice, they look quite normal in relation to their gross organ development, and they produce normal size of litters. And despite that, they have slightly reduced weight. The more obvious when they're getting old, they have a quite normal lifespan. So back to the previous question. So how about their immune cells? So the first step we looked is in their spleen, so looked in their peripheral lymphocytes. And we found that there's a particular defects in their B cells and T cells in their spleen, whereas the rest of other immune cell types that we looked, including eosinophils, neutrophils, monocytes, or myeloid cells, they look quite normal. And this immunodeficient phenotype in those mice probably prompts you to wonder, so is there any human kind of equivalent of those mice? And the Ki67 population genetics is very under-investigated. And also, as you can see, examples here, the frequency of variants in human population is very rare. And so far, there's only one heterozygous carrier found in South Africa and no homozygous found yet. And there was no evidence of haploinsufficiency. And we also found that similar in our mice. So the heterozygous mice do not have a phenotype. But our collaborators found one case of that human Ki67 deficient human patient. So that is a boy and when he was present at the clinic, he was 11 years old and with this rare peritoneal tuberculosis. And I guess that's why our collaborators decide to sequence patient genome, because it's not common that you have tuberculosis in your peritoneal. And what they found, and also confirmed by Sanger sequencing, is that this patient is homozygous for a Ki67 point mutation that's at G, from G to A. What it results in is a stop codon gained and at the arginine 1598 position, so that this creates a frame shift so that the patient has a disrupted Ki67 expression, which is confirmed in the T cells isolated from the patient's PBMC. So our collaborator put the T cells with the stimulus under C3, C2, C28, and looked at the Ki67 expression in the stimulated T cells. So compared to those three controls, our patient had really low Ki67 expression, even in their stimulated T cells. And in line with our mouse phenotype, the patient with the Ki67 deficiency also had quite low numbers of T cells and B cells in their blood. So everything indicates that Ki67 plays a role in lymphocytes. But exactly how, we don't know, or we didn't know yet. But I'll tell you a bit more. So back to the original papers that discovered the Ki67 cellular biology function. We think that if your chromosome look like a clump during mitosis, there might be some defects in the cell proliferation caused by Ki67 deficiency. So we looked in our mouse cells first. We purified splenic B cells from our Ki67 knockout mice and cultured in vitro for 3 days. And these B cells are labeled with cell trace violet, so that the dilution of them indicates cell proliferation. And as you can see from here, compared to Y-type B cells, their CTV dilution looks quite normal, as quantified here. So they can divide as many times as a Y-type B cells in vitro. And also the cell numbers at each peak of the CTV looks no difference compared to the Y-type B cells. So we think Ki67 knockout B cells, at least, proliferate normally. And our collaborators did the same thing with the human T cells. So they cultured human T cells again from the healthy control and our patient PBMC and stimulated again with those T cell stimulus and look at the CTV dilution of the patient cells compared to the healthy controls. And as you can see here, so the red dot here does not mean 2 patients, okay? There was only 1 patient. This is a technical replicates. So compared to the control T cells, our patient still has quite substantial CTV diluted proliferating cells, which indicates that the human Ki67 deficient T cells also proliferate normally. And based on all the previous studies I've mentioned, they also looked at proliferation in their cell lines, and they saw all the cell lines deficient for Ki67 proliferate normally as well. So all of those evidence points to that Ki67 does not play an important role in cell proliferation. So how indeed our Ki67 deficient mice have defects in their lymphocytes then? That's why we looked at the lymphopoiesis. And the early lymphocyte development is a very well-controlled process. As you can see here, they have very distinct stage of development, both in the B cell lineage and in the T cell lineage. And each development stage, they need to conquer some checkpoints. For example, here, from pre-Pro-B to Pro-B, they need to join their D2J segments. From Pro-B to large pre-B, they need to recombine their heavy chain VDJ recombination. So we looked at those early lymphocyte progenitors. So we found a substantial decrease in the B cell compartment in the bone marrow, starting from the large pre-B and the subsequent stages. And similar things were found in the T cell progenitors in the thymus that starts from the double negative 4 stage and onwards. And actually, if you do differentiation index calculation, you'll see that the defects actually started already from the transition of Pro-B to large pre-B in the B cells and from the DN3 to the DN4 in the T cells. So we see that there's a partial blockage in the Ki67 deficient mouse in their lymphocyte development stage. And we know that the proliferation of those Ki67 deficient cells is largely normal. So what is the mechanism that causes these partial blockage then? Again, I have told you that a group found that Ki67 is important for maintaining heterochromatin structure. So we think maybe the genome structure in those Ki67 deficient mice might have some defects that could subsequently cause a defect in their gene rearrangement, and that might have an impact on our B cell development. So what we did was to use ATAC sequencing to measure chromatin accessibility in different stages of B cell progenitors, specifically the pre-Pro B cells that does not have VDJ rearrangement started yet. And in the Ki67 knockout mice, they do not have any defects. And the second population is the Pro B cells. So at this stage, they start to have rearrangement in their heavy chain. And in our knockout mice, this is the stage where the defects start to happen. And then a small pre-B cell population where they have the light chain rearrangement, where they also had quite significant defects in our knockout mouse. So here, this plot showing you that in the pre-Pro B cells, probably as expected, there were not many changes in the accessibility in the regions across the whole genome. But in the Pro B and the pre-B cell stages, you can see quite significant changes in their chromatin accessibility stage compared to the wild type cells that we sought and sequenced. And if you look at those regions that done both in the pro-B and the small pre-B cells and do a geo-ontology analysis, you found that there are quite a few things related to cell differentiation and structure development and those kind of things, which is not surprising. But if you dig deeper in those regions, we found that very interestingly, there is a putative enhancer at the reg locus that was having reduced accessibility in the KSX7 deficient cells at the pro-B and the small pre-B stage. So this links nicely and gives us a hypothesis that maybe the KSX7 deficiency causes some altered chromatin accessibility in their genome, which affect the reg locus expression. So we want to validate that, and we did qPCR measuring the RNA's expression of reg1 and reg2, and we found that in pro-B and small pre-B cells where they should express highly reg expression in those cell stages, but in the knockout cells, they have about twofold decrease in their reg1 expression, although the reg2 was not affected. So does that actually affect the VDJ rearrangement then? So again, using qPCR, we were trying to measure the germline sequence between the D segment and J segment in the pro-B cells, and later on the V and the joint sequence between V and D, because in a normal situation, from pre-pro-B to pro-B, you will join the D2J segment so that you will lose the joint sequence in the pro-B cells. And what we saw in our pro-B cells in the KSX7 deficient case is that they have a relatively higher level of the germline D2J joining sequence, which indicates that this is not happening exactly in the knockout cells. Instead, there are some cells that still bearing this germline D2J joining, indicating a deficiency in their starting of their gene rearrangement. And if that's true, then that will either get carried over into their large pre-B cells, so that they will have some cells that still bearing the D2J germline sequence and also have the V2D germline sequence that's not rearranged. And also if the V2D rearrangement is really affected, then they will have a higher germline sequence as well. So we looked at the large pre-B cells and we saw exactly they have the KSX7 deficient cells still have a higher level of V to D germline sequence, which means that the recombination is less efficient in those KSX7 deficient cells. So lastly, what we did is to see whether if we skip over all the rearrangement, what will happen. So we utilized a mouse model that is reg deficient, so they will have no rearrangement at all, and they bear a pre-rearranged Ig heavy and light chain, so that they're forced to go from pre-pro-B to immature B cells. And we crossed those mice to our KSX7 deficient mice and looked at their pre-pro B cell numbers and immature B cell numbers. So as you see here now, once you don't need all those rearrangement process, the KSX7 deficient cells can still yield as equivalent numbers of immature B cells. Whereas in the previously Ki67 knockout cells with no rescue, it was 10-fold lower. So the B cell development can be rescued by those pre-rearranged Ig heavy and light chains, which again indicates that Ki67 plays a role in gene rearrangement. So to summarize, we found that Ki67 is needed for maintaining proper chromatin structures, and that also linked to maintaining proper gene expression, which then maintains the whole gene rearrangement in lymphocyte development, as we would see in a normal situation. So just one thing for your take-home message is that beyond proliferation, Ki67 also plays a role in lymphopoiesis, and it links nicely the chromatin structure with gene rearrangement in the lymphocyte development. But story is to be continued, because you may wonder, how about those patients still? Like, do they have normal immune response? How does the mature lymphocyte look like? So we are still collaborating with the Casanova Lab at the Rockefeller University and working on those aspects together. And hopefully there will be some more data to show next time. And with that, I would like to thank our present and past lab members at the Immune Memory Lab, although I didn't talk anything about immune memory today, but hopefully next time. And also our collaborator at Austin, Weihai, and Kim's lab helped us doing ATAC-seq, and our collaborator at Rockefeller University. So thank you very much.
Amazing, right on time. Any questions for Zoe after that super interesting talk? Maybe I could start. Oh no, I don't have to. No.
I was just wondering, so it's basically a RAG1 heterozygous sort of for expression. Do you think that's sufficient to explain the six-fold defect in the D to J, or do you think it's more around the accessibility of the RAG as well?
So in human, RAG heterozygosity actually results in a phenotype like partially immune deficiency. So we think that our Ki67 mice probably is mimicking such kind of phenotype. So I think partial RAG1 deficiency probably can explain our phenotype. However, because they have so messed up chromatin accessibility, so there are other regions been involved, which we haven't really investigated. We have looked at PAX5, which should be quite important for B cell development, for example, but expression level was not changed in those knockout mice. So the ones that we looked by qPCR for gene expression, RAG1 was the only one that was having the twofold decrease. Yeah.
Curious, these adaptation, which is like gastrointestinal, any like cells that are quite rigid?
Yeah, so it would be really nice to get more materials from the patient, but the reality is that they have lost contact with the patient, so we can't get any material from them. But yes, we could potentially look in our mouse model and put them on, like looking at the non-lymphoid cells would be quite interesting, because so far what we saw is that only lymphocytes been affected, and the mouse like magically grow quite okay. So the logical step for next is to look at, say, is the chromatin accessibility of other cells normal or not? And could that explain maybe the lymphocytes are the most sensitive ones to a disrupted chromatin organization? Yeah.
Right.
Yeah, on that, I'm just thinking other times when chromosomes sort of mix around, and I'm thinking about fertilization. I'm wondering whether there's a restricted crossover there, but with an inbred mouse, pretty hard to test this at the top. You lost contact with the person. Could be a clone of one of them. Yeah, that was my question.
Okay, you answered it yourself, apparently.
There's lots of things that go on in a mouse that seems to be normal in most.
Yeah.
It's really weird.
Yeah, we really fascinated by that, and I couldn't give you an answer why they are normal, and except that we may just keep looking at our cells, like other cells rather than lymphocytes.
Is it more clonal within the mouse though? Do you not do as much?
Yeah, so we haven't looked at the repertoire per se. We looked at by qPCR again, the gene rearrangement family, like the usage of those proximal or distal family. We found a slight bias towards the proximal family, but it's like whether it's strong enough evidence to say they might have a preferential clonal or not, I can't say.
So I'll just follow up on Paul's questions about the clinical phenotype of the patient. So is there evidence of antibody deficiency, for example? Because of course this is presumably an immunodeficient patient, or any other infections aside from the peritoneal TB?
Yeah, so very good question, and that's also what we are interested in. We asked our collaborators to measure antibody titers, and so far they measured total immunoglobulin levels and some vaccine-related antibody levels, and as far as I can see, there were no deficiency in the patient, which is also curious. Like it seems to have a very big defects in the B and T cell numbers, but all the immunization seems to be fine.
Last question.
Just following directly from that, do you see that in your mouse model as well?
Yeah, so I feel like people are trying to dig out what my second paper gonna be, but this is exactly the direction of our second manuscript, which is under preparation. So we saw substantial defects in primary immune response in our mouse model after one immunization. So currently I'm doing prime boost to see whether that actually can mimic back the patient phenotype.
So we might have to call it quits there. You'll be able to talk to Zoe further over lunch, and thank you for a great talk and pointing out how we're now the world-leading lab on KO67. And thank you also for showing the before and after photos of me. Great pleasure now to introduce our second speaker, Danika, who is a recently minted lab head in the Department of Immunology. She was originally, well, I don't know what originally, but at least in her academic career, an undergraduate at the University of Adelaide, PhD at Zhoujie, having crossed the various deserts and Great Divides to reach Melbourne, leaving in 2015, I think, to go to the Babram Institute to do a postdoctoral fellowship with Michelle Lintemann. And I think it must have been in your PhD that you developed a fascination with intractable immunological problems in humans, but you then pursued, working on malaria, but you then pursued both at the Babram and then even further here. So Danika returned from a very successful postdoc with Michelle to start to join my group in 2020, and then became a lab head in 2022, three, somewhere around there. Everything's a blur. With a focus, in fact, since she returned, as a recipient of the Michelson Prize from the Human Immunome Project, a very prestigious award, which only one or two are given out per year, to pursue complex problems in human vaccinology. And her choice was an incredibly complicated one to work on asparagines and the immune response to it, and why it's been so difficult to develop vaccines. So it's a great pleasure to have you present your work and what your approach is going to be to resolving this problem in the next 2 years, I guess. Yeah, thank you very much, Dan.
Thanks, Dave. That's a bit of a tall ask, but we'll give it a red hot shot. Thanks. It's a pleasure to be here today to give a kind of a flavour of the work that me and my team have been doing since I started my lab a little bit over a year ago now. So I just wanted to start by making some disclosures. I have received some financial funding from a US biotech, Dynavax, but that's unrelated to the research in the seminar. I do have quite close working relationships with several Strep A vaccine developers, but they haven't influenced the findings or conclusions that we're making from this research. So Group A Streptococcus, Strep pyogenes, or what I'm going to refer to it today as Strep A, has a really broad spectrum of diseases ranging from acute through to chronic. Typically, it manifests as strep throat or tonsillitis, but can also cause skin infections called school sores or impetigo. It can kind of get past that mucosal tissue and cause systemic diseases, things like scarlet fever, and some really nasty invasive infections, things like toxic shock or necrotizing fasciitis that have up to 60% fatality rates once you present with that condition. So across all of these, it's about 800 million infections happening around the world every year and 150,000 deaths. Strep A infection can also trigger some really fascinating post-infectious autoimmune sequelae, things like glomerulonephritis and rheumatic fever. And there's about 600,000 cases of ARF every year, and also some fairly quirky neurological sequelae as well. And it's these two ones, the glomerulonephritis that can trigger chronic kidney disease and rheumatic fever causing rheumatic heart disease. So there are around the world 34 million people living with rheumatic heart disease and over 340,000 deaths. So these are just really, really staggering numbers. And sadly in Australia, our First Nations populations have 60 times the rate of non-Indigenous Australians. So we've had an effective antibiotic against Strep A since for over 80 years in the form of penicillin, and most strains to this day are still susceptible to that antibiotic. But despite having essentially a cure, we still have those staggering numbers of deaths happening around the world every year. And it's really appreciated that the only way we'll achieve a sustained reduction in the burden of Strep A diseases is through a vaccine. What I find fascinating is we did have one. In the 1920s, these two pioneers, George and Gladys Dick, literally wrote the book on scarlet fever. It's estimated they immunized tens of thousands of adults and humans with a fairly crude preparation of culture supernatant that if you gave 5 doses, you could protect against scarlet fever. And just amazing work controlling outbreaks of scarlet fever with their vaccine. As you can imagine, receiving an immunization of a culture supernatant chock full of toxins and all sorts of other things wasn't particularly well tolerated. And so it was really kind of superseded with the widespread use of penicillin around the 1940s. But the interest in a Strep A vaccine did pick up again in the 1960s, but it kind of really ground to a halt at around 1969, where this particular M protein vaccine was implicated in triggering rheumatic fever. So a vaccine causing the disease that it's aiming to prevent. And that led the US FDA to essentially put an embargo on vaccine trials that lasted for about 30 years. And that only lifted in 2006. But that's 20 years ago now. So what's the holdup? So in this graph here, you can see the amount of R&D funding around the globe plotted against the amount of disability adjusted life years for each condition. And you can see rheumatic heart disease right down there at the bottom. Huge daily burden, really little global R&D. So long-term neglect from big pharma. But things are changing. There's been recent global momentum from a lot of big pharma companies to begin to be interested in this space. There's a range of vaccines that have either just entered for Griffith University. They've just entered phase 1 trials. But there are a lot of vaccine companies gearing up to enter clinical trials. But suffice to say, there hasn't been a full commitment yet. There's a lot of kind of apprehensiveness about commencing these vaccine development programs. And part of that is because there's just such a limited knowledge around immune responses to this pathogen in terms of how our bodies respond when we naturally encounter it, but also what a vaccine needs to do. And that's partly because it is a human-restricted pathogen. There are animal models, but they're not great. You know, considering these animals physically are much, much smaller than humans, you need either 100 to 10,000 times the dose of bacteria. So that's not going to mimic both the way in which our immune system sees these bacteria, but also, you know, it's not going to pick up the full picture of the virulence of the bacteria that has evolved to target humans. And so that means that on the human side of things, the immune response is really still a black box. And there's very little research done past just measuring serum antibodies alone. And so there's big questions that remain open in the field, including, can immunity from natural infection be protective and long-lasting? We don't know that. We know that epidemiologically, children have high incidence and that drops off with adulthood, but we don't know if that's due to protective immunity or not. And it's likely, I believe, not the case. So that really leads the question, well, what does a vaccine need to do in order to protect us? So this is a schematic showing just a simplified version of the clinical trial pathway from phase 1 through to registration. And I've just highlighted, you know, these various different hurdles along the way. The first being safety. The unique thing about Strep A vaccine development is that risk of rheumatic fever and that sort of historical concern that a vaccine could trigger the disease we're trying to prevent. And that continues to this day because the definitive molecular trigger for rheumatic fever is still unknown. Whether or not that is due to molecular mimicry, some bacterial molecules that mimic host tissue, or a bystander effect of the tissue damage that happens during infection remains unclear. And the second part of that is which of these antigens are going to be immunogenic in humans. So it's likely that the first trials will happen in healthy adults, but the target population is likely young children or infants. But they really represent two very different categories of exposure. So adults have probably had estimated at least 5 encounters with Strep A throughout our lifetime, but this could be much, much lower. And that really is important to think about in the context of giving a vaccine, that you could have that pre-existing immunity there that could influence your vaccine response for better or for worse. And so those are some of the issues that my lab's trying to tackle. And I just wanted to kind of highlight these 4 different regions that we're trying to work on in the Strep A space. Trying to understand the human response to infection, trying to understand some of those autoimmune sequelae. So this is a new collaboration we're starting with Telethon Kids Institute and the Menzies to look at rheumatic fever in First Nations children. To understand how superantigens in invasive infections can perturb the immune response, and really in future to be at the forefront of these vaccine development and trials that are going to be happening in Australia. In the talk today, I wanted to highlight some of the great work my team and I have been doing over the past few years to tackle the infection side of things. And firstly, I wanted to start by showing some data, which is serum antibody work from a human challenge model. So a few years ago now, Andrew Steer and Josh Osowicki from the Murdoch Children's Research Institute established the world's only human challenge model for Group A Strep. And what this means is healthy adults were recruited and had Group A Strep bacteria painted onto their tonsils. 3 to 5 days later, 19 of those, so an attack rate of 80%, went on to develop pharyngitis. And we have biological samples from pre-challenge all the way out to 3 months. And this is a really unique opportunity to ask what pre-existing immunity is present before infection. Because typically, most studies out there, they only take blood samples at this sort of acute point of infection. The second question we can answer is what antibodies are actually elicited by infection. So Strep A is a very complex bacteria. It's got over 2,000 proteins. So we had to use a kind of multi-antigen approach. So showing here, we've picked 17 of the leading vaccine candidates to study. The picture we saw from first looking at things was really, really complex. So this is scaled ELISA titers for saliva and IgG, pre and post challenge, split into those that didn't develop pharyngitis. It's clear to say we couldn't detect a picture, and that really reflects the kind of complex past exposure with Strep A. But we could start to detect some patterns when we normalized each person to their own baseline. So showing you here for these 10 antigens, the fold change pre and post challenge in saliva for IgA and serum for IgG. And some of these antigens here, which you can see were significant, are among 3 of these leading vaccines. So that's really good news, I think, that these antigens, at least in the context of natural infection, are immunogenic. So, potentially getting over hurdle number 1. Obviously, a human challenge model is quite a contrived setting in which to get an infection. And this is a human challenge of adults. And obviously, for a vaccine to work, it needs to work in children. So how much does the human challenge response mimic natural pharyngitis? So shown in red here are the human challenge adults. And this is compared here for these 2 antigens that are used clinically to define strep pharyngitis. And this is cohorts of children from New Zealand. There are different children because they're not paired here. So healthy children and those that had a pharyngitis episode confirmed a month previous. And what you can see is kind of the magnitude of change pre and post pharyngitis mirrors what you would see in children. We were really encouraged to see that the antibody responses we detected in blood 1 month after challenge occurred quite quickly and were maintained out to 3 months post-infection, sort of really supporting that they're not just transient antibody responses, but suggesting that there could be some sort of long-lived immunity component to this. But interestingly, when we sort of tried to dig into what are those individuals that are responding, we saw this inverse correlation between the amount of antibody you mounted in response to challenge, so the fold change over baseline, and your pre-existing IgG titer. So those that had high levels of antibody to this particular antigen before challenge actually mounted very little antibody in response to challenge. We're sort of digging into this more mechanistically at the moment, but what we think it could suggest is that either this pre-existing antibody can somehow mop up the antigen, causing it to be cleared away, preventing the initiation of de novo new B-cell responses, or alternatively could be masking epitopes such that we can't generate new antibody responses. I guess it's worth noting here, you'll see that if you remember, the attack rate of pharyngitis was 80%. So we know that these antibody levels that people have pre-challenge, for the most part, are not protective. And so what this suggests is that there could be some level of inhibition from pre-existing antibodies that a vaccine may need to overcome in order to be effective. Hopefully with a strong adjuvant and a big dose of antigen, this won't be a problem, but I think it's cause for concern and definitely indicates the importance of understanding that pre-existing immune memory. Those who may have keen eyes may have mentioned that may have seen that most of the blue individuals are kind of clustered up here, showing high levels of pre-challenge IgG. So then we wanted to kind of dig into, is there anything about their serum antibodies before challenge that might influence their outcome? So we took all of the 37 variables that we'd measured pre and post challenge and took 2 complementary approaches to dig into this. The first was taking a spectrum of clinical variables and just doing correlation analysis. And we found these 10 antigens here that showed at least one correlation with these various different clinical variables. And they broadly grouped into this first group here, where high baseline antibody seemed to associate with early onset pharyngitis. And the second group, where high antibody at baseline seemed to correlate with no mild or delayed pharyngitis. The second approach we used was using sparse partial least square discriminant analysis. So essentially you build a model where you give the outcome variable and ask it to find the variables that best explain those different groups. And when we ran this model, it basically said that these 5 variables here best explain the difference between the pharyngitis and non-pharyngitis participants. So again, higher levels of TF and ADI for the pharyngitis group and these 3 antigens here. So this is the raw data for those 5 antigens. This is baseline antibodies, and you can see this skew towards having high levels for these 2 antigens in pharyngitis participants, and a hint that, you know, these 3 antigens here are higher in those that don't go on to develop pharyngitis. So at this stage, with, you know, the 6 individuals that we have in this first challenge, we don't know yet whether or not this could represent sort of a immune correlate of protection, but it does suggest to us that the antibodies from your prior infection may have an influence on how you respond to infection. So just to quickly summarize the human challenge aspects, we do see highly heterogeneous antibody responses in both their magnitude and specificity, but we are seeing responses to some of those key vaccine antigens. That baseline antibody may influence susceptibility, and that pre-existing humoral immunity may blunt memory recall. And I think that really emphasizes the importance of understanding what immune memory is there beyond serum antibodies to the actual cells that will be maintained long term. So when I moved to Monash in 2020, was started this Group A Strep project, and really there were very few tools in the field to understand Strep A immune responses. And really, you know, in our blood, we've got B cells, but there's only a very, very small fraction of those that will be specific to any one antigen. So in order to tackle that for Strep A, we needed to build some new tools. And the way we did that was basically making these baits, these proteins that we couple to fluorescent streptavidins that bind to the surface of specific B cells. So we can detect them using techniques like flow cytometry. To get better specificity on that, we combine this by making the same antigen on 2 different colors. That's what we called dual discrimination approaches, and it'll be shown as a kind of diagonal line in a lot of the plots I'll show you coming up. But as I mentioned, Strep A is a very complex bacteria, so we had to develop multiplex approaches to do that. And now typically we're running up to 16 tetramers in a single panel, getting the most out of the precious clinical samples that we have. We're also combining the tetramers with oligotags such that we can retain the antigen specificity in downstream sequencing approaches. So this is some flow cytometry plots of B cells showing what these multi-antigen approaches look like. So here we have all 5 of these antigens are conjugated to APC, and then these different streptavidins can distinguish the cells that are specific to here for HA, for influenza, and these 4 Strep A antigens. And what our analysis of human blood donors has shown so far is that most people, most adults, have immune memory against all of these vaccine candidates. And that really suggests that vaccination is going to be a secondary response. We've also been looking kind of further from blood into human lymphoid tissues. And this has been a wonderful collaboration with Michelle Boyle's lab at the Burnett, through Stuart Tangy at the Garvan. So some human spleen samples from organ donors, where here we've got influenza hemagglutinin again, and just a representative Strep A antigen SCPA on the diagonal here. And in all of the spleens we've looked at so far, we can find Strep A specific memory B cells in the periphery, I guess, ready to guard against infection of the tonsil or protect against things like scarlet fever. You can see that the kind of magnitude here compared to the flu response is lower for some of the antigens, but not all. So this is work that's kind of ongoing. And the approaches we're kind of using to dig into this is single cell sequencing, where we can not only get information about what sort of subsets of B cells are there, what isotypes they express, but also from kind of each of the dots here represents a cell. We can study the B cell receptor that those express and then make monoclonal antibodies. And this is going to be a really powerful approach for both understanding immunogenicity, immunodominance, but for a lot of these vaccine candidates, we don't actually know what the immune system needs to do. We don't know if the immune response that we can mount in response to infection or then potentially vaccination would even be functional. So we think making monoclonals is going to be a really powerful way to do that. This is just two little vignettes of these 5 monoclonals that we've made that have various different levels of affinity against their antigen, and we found can be functional in vitro assays with monocytes. I've kept things a little bit vague here in terms of the antigens and antibodies themselves, because we're in the process of preparing a invention disclosure with Monash Innovation that Andrew Bray's been helping out with. So obviously I've shown you blood and spleen, but we get strep A infections in our mucosal surfaces like our tonsil. So we've also been trying to understand what's happening at that site of infection using tonsil samples that get taken out for obstructive sleep apnea. So very grateful to collaborators at both MCRI and Michelle Boyle's lab at the Burnett that have helped get us access to some of these tonsil samples. So this is very much a work in progress. We're still building up the kind of sample sizes so far, but we have the cohort from Burnett. We actually don't know the strep A colonization status, but from MCRI, we do know whether or not they were or weren't colonized. But you can see again, in all of the tonsils we've looked at so far, we can see immune memory against all of these strep A antigens. Just to kind of extract one example here, it's low numbers of samples, but we do see this greatly elevated number of antigen-specific cells if there's an active colonization going on. So these tonsils we don't think are they don't have tonsillitis, so in kind of active pussy infection, because that would be a contraindication to the surgery taking place in the first place. But we think what this is representing is a sort of transient state of colonization, which happens a lot in humans. But really kind of arguing that it's not a kind of immunologically silent process, that there is a very dynamic response to that infection taking place. And another piece of evidence we have for that is that if we look at these cells in a colonized individual, they express markers, CD95 and CD38, that distinguish germinal center B cell responses. So this is a very dynamic process that happens in response to infection. And what's really fascinating for me as an immunologist is that this is actually the site at which evolution of that B cell response is taking place, and the B cells are actively undergoing mutation of their B cell receptors. So I think this is really promising, again, because it suggests that these antigens that people are interested in exploring for vaccine purposes are immunogenic, and the fact that we do see evidence of a germinal center response to infection suggests that at the fundamental level, these antigens have enough epitopes to support durable responses, suggesting that we could achieve that goal through vaccination. And again, we're kind of really actively studying these responses to understand whether or not the responses that we do make, if they can be kind of antibacterial. And so the next steps for this work over the next few years is to, I'm sorry, I didn't mean to animate this. Let's click through that. Is to kind of understand the responses happening at the tonsil, combining that with analysis of B cells and T cells as well in the human challenge, to really kind of nail down immunogenicity and function for these vaccine candidates. So that's to kind of get at vaccine efficacy. On the other side of things, obviously there's still that vaccine safety concern for these strep A antigens. So again, working with cohorts of children with rheumatic fever to look at these vaccine antigens and at the same time look at the autoantigen response to understand, is it molecular mimicry happening? What are the molecular drivers of rheumatic fever? And are these vaccines going to be safe? And just to have open positions for any interested honours and PhD students. So that just leaves me to thank all the people involved in this work. Particularly want to shout out to Kathy Pitt and Holly Fryer. Kathy is an amazing RA who has been completely instrumental for getting all of these technologies off the ground. Holly joined my lab at the start of the year for a PhD and is already hitting many home runs. I've had a lot of support from David since joining the department. Wouldn't be where I am without that, so thank you. And obviously this is human clinical research, so a lot of collaborators both within Melbourne at the MCRI and at WeHi, but also around Australia and around the world. And I'll stop there and take questions. Thank you.
Thanks, Danika. As you said, a very, as I said, yeah, very complicated problem that you're addressing. So time for a couple of questions if anyone has any burning issues. Yeah, Matt.
That was great, Danika. I was wondering why 5 encounters with strep A is not more immunogenic. There's a lot of antigen variability. And secondly, so I really like the data showing the immune protection to pharyngitis, but do you know any of the limits from that kind of determine what turns pharyngitis or scarlet fever or tiger into full-blown rheumatic fever?
Yeah, really good questions. So to answer the first bit, there are over 200 strains of Group A strep, and that's linked to immunogenic protein on the cell surface called the M protein. So that is one of the ideas around why immunity might be not that effective. But also we know that protection against homologous infection is also not great. And part of that is because the bacteria has just evolved to get around our immune system. Like a lot of these vaccine candidates are also virulence factors that specifically target complement cytokines. It's got the super antigens to kind of misdirect the T cells away from the bacteria itself. So I think that has a big part to play. It's just got a huge arsenal of virulence factors that subvert the immune response.
So I guess a lot of the vaccines have a lot of bases covered then in terms of...
Yeah, so kind of historically people were trying to target the M proteins, but like the most advanced one went to 32 valent and that wasn't broad enough. So that really becomes a challenge for kind of protein-based vaccines. So there's a shift towards these more kind of multi-component conserved virulence factor approaches, where the thinking is if we can kind of cripple some of those virulence mechanisms, then that might give the immune system the chance to both, prevent invasive infection, but then, get to protection is the hope. In terms of what the trigger is for rheumatic fever, there's evidence out there showing that skin infections are important, throat infections are important. Whether or not you ever need the infection to get past the tonsils into the bloodstream and cause things like scarlet fever in order to get the autoimmunity, I'd say it's not particularly clear, although clinically a lot of children that present with ARF have never even had antibiotics often. So that would kind of suggest that they haven't got sick enough to precipitate needing to present at hospital or something like that. So I think there's studies showing that it's the higher the incidence or transmission of Group A strep, that if you've had more exposures, that puts you at greater risk. It's a little bit of an unknown. Yeah.
One last quick question and one last quick answer. Frank, yeah.
I suppose whenever you hear the word autoimmunity, you always think Ben like C. So anything known around that, susceptibility or protection, I suppose?
Yeah, it's a good question. There's AGWAS looking at rheumatic fever. it hasn't been replicated in the subsequent 2 GWAS for MHC. So not really looking like that's a problem. Well, not the solution, I think. Maybe some signals coming up for BCR locus, but yeah.
Been around for a while. Thank you very much, Danika, and thank you again, Zoe. Fantastic talks to both of you. I apologise to anyone on Zoom who might have had a question. There's no way I'm going to be able to work that out. So please, could you join me in thanking our 2 fantastic speakers today, and then join us for lunch. I'm not. Yeah, there's usually like a little hand up or something over here.
Hi.
I'm gonna mouse over there.
Oh, right. I don't know that. But let's delete this hit.
Yeah, we expect it to walk there are they have helium? Can you charge or something?